There's no questions. Let's begin. Today, we're actually supposed to be starting chapter 2. We're a little bit behind schedule. So we'll finish chapter 1 and then move on to chapter 2. Do note that you have a practice quiz today, which is due, I think, this Wednesday. It's practice quiz 00. Let me see if I can pull that up. Don't really need this. You should take the practice quizzes because they'll give you practice for my quizzes. And they have questions similar to what you'll find on the quizzes, although none of the questions will be the same as the questions on the quizzes. So don't use it to study, but just use it to determine how well you know the material. So we got an image for our course site. So hopefully you're seeing my screen. Can somebody confirm that you're seeing this?
Yes.
Thank you. So there's a practice quiz 1A, which gives you short answer questions or essay questions, and a practice quiz 1B, which gives you multiple choice questions. For practice quiz 1B, you must download Respondus Lockdown Browser and then use that browser to run the practice quiz 1B. That quiz cannot be accessed unless you're using Respondus Lockdown Browser. And you should download it so that you can take the future multiple choice quizzes and make sure that Respondus Lockdown Browser is working on your system before you take the quizzes. So that's another reason why you should take the practice quiz, to make sure Respondus Lockdown Browser is running on your system. If it does not run on your system, and that happens about in one student every other term, contact the computer help desk. They're very good at getting it up and running on your system. Usually you just need to go in and change a setting on your computer. Any question about any of that? All right, if you take these practice quizzes, I will give you extra credit points. I think it's 1 extra credit point for each quiz. So each question is worth about 1 point, or 1 point per extra credit quiz. Any question about any of that? All right, let's go back. Let me mention, I'm not going to show you the grades, but I did a lot of grading. So I did grading for the worksheet for the lecture, and then I graded the 2 labs. On the lab grade, you either got 5, meaning that's a complete grade for both labs, or you got, what is it, 0.01 points. The 0.01 point is not your grade. That's a code for me to realize that you didn't get 100% on the lab. And on the first grading, I will go back and regrade it. So you have until Saturday to submit corrections if you did not get 100% on the lab. And I will then go back and regrade the lab. And then your grade will be graded in the normal way, meaning it'll be someplace between 0 and 5 points. But on the first grading, you'll either get 5, meaning 100%, or you'll get 0.01. And like I said, that's a code to tell me to go back at that assignment and regrade it if you submit a correction. Any question there? Oops, sorry, I don't have this on. Let me get this on. I had it even plugged in. I just didn't have it on me. Can you guys hear me better now?
Yes.
Okay, great. All right, so you do have a second chance in the lab. And please understand that's exactly what it is. I consider the lab a learning experience. I will tell you which questions are wrong or partly wrong, and then you can go in and resubmit the lab a second time. Any questions? All right, I think I'm the only instructor that does that for the lab, so I'll consider it a bonus for taking my class. Anything else I need to talk about? I don't think so. So let's move on because we were a little behind schedule. Any questions? So is this the last slide we talked about? Pasteur's experiment disproving or did we go on to this slide? Which slide did we end? Hello? I've got two markers here, so that's why I'm a little confused.
We ended on the one of the theory of biogenesis with the beef or the broth.
So you're saying we ended with this slide?
Yes.
Okay, thank you. Yeah. Come on in here and delete that. All right, let's see if I can move that up a little. There we go. The golden age of microbiology began with Pasteur's work. The discoveries coming out of the golden age was the relationship between microbes and disease. And this led to the germ theory of disease. The disease was caused by germs or microorganisms. This further led to the development of the concept of immunity. We have an immune system to protect us from diseases or germs. And then that led to antimicrobial drugs that we could use to treat germs causing disease. Some of the important events that came out of the golden age of microbiology. Pasteur developed the basic idea of aseptic technique, where you can take a culture, grow it as a pure culture, and then aseptically transfer it into another culture, another dish. Koch further developed that by making a technique for streaking for colony isolation. And this was a big event because he made an easy way to get a pure culture. It was possible. In fact, Pasteur did work with pure cultures, but his way was very difficult to get a pure culture. And Koch just simply streaked it out on an agar dish, and he was able to easily get pure cultures. And we'll talk a little bit more about that in the lab. And then another development was Hess, who was an employee of Dr. Koch, developed nutrient agar, a very simple and easy nutrient to grow microorganisms. And this was the first general purpose media we talked about, nutrient agar. And then about the same time is when Sergei Winogradsky developed the idea that microorganisms were involved in the biogeochemical cycles on the planet Earth. And we talked about the nitrogen cycle, where nitrogen recycles or moves around in the ecosystem because of microorganisms. But that also happens with other nutrients, and Winogradsky studied it with the sulfur cycle as well. Let me go back just a little bit. Why Winogradsky was able to discover this was he was unlike the other early microbiologists who, to study their organism, they got a pure culture containing one species and then studied it. Winogradsky studied ecosystems containing multiple microorganisms, and that's why he was able to study the biogeochemical cycles on the planet Earth. Pasteur showed that microbes are responsible for fermentation. Alcoholic fermentation is conversion of sugar to alcohol and is used in the making of beer and wine. And Louis Pasteur showed that it was microbes that were causing the fermentation to occur. He also showed that microbial growth is responsible for the spoilage of food. And he showed that bacteria can use alcohol and then produce acetic acid, spoiling the wine or the beer, fermenting it to vinegar, acetic acid, making it not very palatable to drink. Out of that came the theory of that microorganisms might cause disease. Pasteur further showed that bacteria could be killed by heat and that he could use heat to kill off some of the bacteria, if not all of the bacteria, but not hot enough to evaporate all of the alcohol in the wine. And from that, he got pasteurization, the application of a high heat for a short period of time to help not really sterilize the food because it doesn't kill all bacteria, but to make the food last longer. And that we call pasteurization, which is used in a wide variety of foods. Pasteur first used it with beer, but today it's mostly used with milk. And that removes most of the bacteria, but not all of the bacteria from the milk. And that increases the shelf life of the milk. And then it also removes the human pathogens from the milk. From the realization that fermentation was caused by microbes affecting organic material, converting the sugar into alcohol, people asked the question, might there be similar relationships with plants and animals? And may microorganisms cause disease? This became known as the germ theory of disease, that bacteria or other microbes could cause disease. This was an important advancement because originally people thought that poisonous vapors were believed to cause infectious disease. For example, they said that somebody got cholera from walking through a swamp and they breathed in the poisonous vapors of the swamp to get cholera. Well, in reality, they probably were exposed to cholera in the swamp and then got cholera while they were walking through the swamp. Any question about any of that? We'll skip the germ theory of disease and read about that. Who proved the germ theory of disease or disproved spontaneous generation, or what's the other one? Well, we'll just say he proved the germ theory of disease, was Robert Koch. And he showed that a bacterium, Bacillus anthracis, actually caused the disease anthrax. And then he provided the experimental steps that were required to show that a specific microbe causes a specific disease. And he wrote this up, and this is known as Koch's postulates. The steps you use to show that a given organism or microorganism causes a specific disease. And we'll run into Koch's postulates 3 times this term It's an important concept, and we'll run into it 3 times. So you're likely to see it on a quiz, as well as probably the final, and maybe even another quiz. Anyways, when he did this, he proved the germ theory of disease because he showed that Bacillus anthracis causes anthrax. And it was in 1884, you don't need to know the name, is when he wrote up the steps for Koch's postulates. About this time, Edward Jenner came up with the first vaccination, where he made a vaccine against the disease smallpox. What he did was he inoculated a person with cowpox virus, and that protected the person against smallpox, against a different virus. This was not understood until Louis Pasteur started experimenting with vaccines. And what Louis Pasteur showed was that cowpox is genetically similar enough to smallpox so that when our immune system makes antibodies and an immune response against cowpox, it cross-reacts with smallpox. And with Louis Pasteur's work, we then knew how to make other vaccines. So Edward Jenner made the first vaccine, but it wasn't understood how that vaccine was working. And so nobody else knew how to make another vaccine. And it was actually Louis Pasteur who showed us how to make vaccines. And the first vaccine he made was against some disease that causes disease in chickens. And what happened was chicken farmers went to him and asked him for help, and he did research and showed that it was a microorganism. I think it was a virus, but I don't remember, that was causing chicken disease. And he made a vaccine for it. And then out of his work, we learned how to make other vaccines, including the rabies vaccine. Louis Pasteur, you can read about it. That was one of his great achievements, was he made a vaccine against rabies. Because rabies back then, if you got the disease, you were going to die from it. There was no possible way to cure a person from rabies. And to my knowledge, there's only been one person in the world who's ever survived a rabies infection. And this person wasn't normal. They got the rabies vaccine, but then later they came down with rabies, and they think the person survived because the vaccine worked well enough that it kept the person alive, but did not work well enough to prevent the person from getting rabies. Vaccination is actually derived from vacca, which is a word meaning cow, and that's directly coming from Edward Jenner's use of cowpox to make the first vaccine. Another concept that came out of this is that the protection that the body makes against disease, we call that immunity. Any question about any of that? From this work, we got the birth of modern chemotherapy. Chemotherapy, initially and strictly speaking, is the treatment of disease with chemicals. However, nowadays we use chemotherapy almost exclusively to mean treatment with drugs against cancer. Okay, but initially that was treatment of any disease with chemicals. And we still use a chemotherapeutic agent, is a drug or agent to treat against an infectious disease. It can be a synthetic drug, meaning man-made, or it can be an antibiotic, which strictly speaking an antibiotic is a chemical produced by a living microorganism, a fungi or a bacteria, that inhibits or kills other microbes. Now, let me state that this is only a textbook definition for what an antibiotic is. When we use the term antibiotic in common usage, even clinicians use the term antibiotic to mean a chemical that protects against a microbe causing disease, meaning most people call both penicillin G an antibiotic and ampicillin or carboxycillin antibiotics because they treat against disease. But the textbook definition of an antibiotic, only penicillin G is an antibiotic because it is produced by a living microbe, a fungi. The others, even ampicillin, which is an old antibiotic or an old chemotherapeutic agent, is a synthetic drug, meaning that humans make it with ampicillin. You don't need to know this. It's actually made by a microorganism, and then humans modify it, so it's a semi-synthetic. Carboxycillin is totally made by humans in the lab. And strictly speaking, those are not antibiotics. Any question about that? No, generally, when I'm using the word antibiotic, I will use it the way that it's used in common usage, meaning ampicillin is an antibiotic as well as penicillin G. I will not use the textbook definition of what an antibiotic is. Any question about that? The first chemotherapeutic agent that was ever discovered was quinine, which was the bark of the cinchona tree. And it was used to treat the microbe causing malaria. When do you think we first had the first chemotherapeutic agent which was used to treat against malaria? When do you think that was? Come on, somebody can guess. All right. Nobody's going to guess, so I'm not going to tell you the answer. Paul Ehrlich, out of this word, came down with the first chemotherapeutic agent that was discovered to treat a disease. And he found salvarsarin, a synthetic arsenic drug, which treated the disease syphilis. And he came up with this in 1910. He called it a magic bullet, a drug or a chemical that could be used to treat a microbe and then cure a specific disease. Any question about any of that? All right. So Ehrlich came up with the first chemotherapeutic agent, which was used against syphilis in 1910. And you don't need to know the year, but you should know Ehrlich came up with the first drug to treat a disease. And what he did was he just simply looked at different chemicals. And that one didn't work, so he tried another chemical. And that one didn't work. And people put on the sign on the top of his door, "Abandon all hope, those who enter here," because they just went on and on and on. And it wasn't until he tried slightly over 500 chemicals that he finally found one that was able to cure syphilis. Alexander Fleming was the first one who discovered the first antibiotic. He observed that the Penicillium mold made something that he called penicillin that secreted out of the mold and then prevented the growth of the bacteria. What he did was he streaked out a plate similar to this, and he was a sloppy microbiologist and he got contamination on the plate. And then he left the plate on the counter so that both the bacteria and the mold could grow. And then when he came back over the weekend, he noticed that he had mold there. And then around the mold, there was a region that there was no bacteria growing. And he correctly concluded that something was coming from the mold, preventing the bacteria. And he called that penicillin. He was not able to clinically purify penicillin. That didn't come until the 1940s. And then once they got it purified, they called it penicillin G, the first clinically found antibiotic. However, it was not the first clinically found, the first clinically used antibiotic. In 1935, somebody made a man-made antibiotic, sulfonamide, which was a sulfur drug. And it was the first clinically available antibiotic that could be used in 1935. Antibiotics, penicillin G was not used until the early 1940s. It became available in World War II, where they were able to mass produce penicillin G and then purify it and then inject it into a patient. If we have time, I'll tell you the story about the first 2 patients who were treated with penicillin G. It's an interesting story. All right, a couple of comments about this lesson before we end it, and that is microbes are involved in human disease. Bacteria were once classified as plants, hence the term flora for microbes. Flora means flora, plants. It's now replaced by microbiota instead of microflora. And if you ever heard me say the term flora or microflora, you can correct me. It's because in my day, when I took microbiology, microbiota was not a term in use yet. It later came into being. And the normal microbiota are the microbes that are normally present in and on a person. And so we all have a normal human microbiota, which is actually important. We'll talk a little bit about how it's important. All animals and plants are host to symbiotic microbial communities which grow on them. And the normal microbiota is central to their welfare. For example, let's see if I discuss that in the next slide. They help prevent disease. We'll talk a little bit about that. They can secrete toxins that inhibit the growth of certain pathogens. We'll talk a little bit more about that later. And then they can produce nutrients and growth factors for their host. Like, for example, we get some folic acid and vitamin K from our normal microbiota, mainly the normal microbiota in our gut, producing folic acid and vitamin K. And then the normal microbiota is very important in our digestion. It helps us digest our food. If we didn't have the normal microbiota, we would have trouble digesting our food. The normal microbiota is normally found on and in the skin, the oral and nasal cavities, the upper respiratory tract, throughout the digestive tract, and in the urethra. The normal microbiota is not normally found internal to the body, such as in the internal organs or the bone or the blood. And if it's there, it's a disease. Okay. The normal microbiota is not normally found in the lower respiratory tract, not in the lungs, for example. And if it is found in the lungs, we will call it pneumonia. Any question about any of that? So the normal microbiota do not normally cause disease. However, they can cause disease in certain circumstances. Let me go back to that slide. For example, Staphylococcus epidermidis living on my skin does not normally cause disease, but if I were to get a puncture wound, I could get Staphylococcus epidermidis growing beneath my skin in the puncture wound, and then that would be an opportunistic infection. We'll talk a little bit more about that later. And then the normal microbiota can cause disease in people who have an impaired immune function, like especially AIDS patients. But normally, the normal microbiota does not cause disease. An infectious disease is one where a pathogen overcomes the host resistance, and then disease results. Infectious diseases are caused by pathogenic microbes, meaning a microbe that can cause disease. An emerging infectious disease is a new or changing disease that is increasing or has the potential to increase in incidence. Like a new and changing disease is COVID-19, and that is an emerging infectious disease. Does anyone know another possible emerging infectious disease? All right, nobody's taking a
Is
guess.
it called like monkeypox or something like that?
Yeah, something like that. Monkeypox was not spreading in the United States until recently. I think it's mainly spreading by sexual contact. It can spread in other ways, but it's tending to spread mainly by close contact. Fortunately, this disease doesn't spread easily, but it wasn't one that happened in the past, and so it's emerging infectious disease. One that has the potential to increase is bird flu. Okay, so that's one that is an emerging infectious disease. It hasn't emerged yet, but it has the potential to increase in incidence, and so they're watching it. All right, that's all I want you to know about emerging infectious disease. I've got some slides on it, but we're not going to talk about it. All you need to know is what an emerging infectious disease is. Everything else I'm not going to test you on. Any question about any of that? If there's no questions, I'm going to end this and begin lesson 2. Let's see, where do I have that? All right, normally when I show you a lesson, I give you a slide that gives you the rough outline, as well as the major goals of the lesson. Now, understand this is only the major goals. If you want to know all of the objectives, you should come up to here to take a look at the study guide where I've got all of the objectives at the top of the study guide. Any question about any of that? All right, while I'm here, let me make lesson 1 available to you. I'll make the PowerPoints available to you. There's actually a PDF file after we finish the lesson. All right, so know the terms. What's an organic and an inorganic compound? What's a hydrocompound? What are polar and nonpolar molecules? What are hydrogen bonds? What's an acid? What's a base? And what is pH? You should also be able to, with respect to the 4 major biological molecules, be able to describe the relationships and the characteristics of the monomers and the polymers. Be able to describe the composition, structure, and function of each of the major biological molecules, and then be able to give examples of each. What are the 4 major biological molecules? They're the main molecules of life. You can find 3 of them on almost all of your list of ingredients on the food we eat. You look on the box of cereal, you'll find 3 of the biological molecules. What are they? Come on, you should have had that in Biology 160.
Glucose.
That's a good guess. Glucose is a monomer of one of the major biological molecules. The major biological molecule are carbohydrates. And you can find that on a box of cereal. What else can you find on a box of cereal? All right. The other
Protein.
biological molecules you can find on a box of cereal are proteins, lipids, and we usually call that fats when we eat them, but lipids is a more general term. And then the 4th you won't find on the box of cereal, that would be nucleic acids, such as DNA. Those are the 4 major biological molecules. You only find those molecules in something that is either alive or was once alive. Like if you find it in a rock, it would be from something that was once alive. However, you would not find the 4 biological molecules like on a rock from the planet, not the planet, from the moon. Okay? Because there was not life on the moon. When we're talking about chemistry, it's important to understand the scale of things, know the relative size of things. Let me see if I can blow this up. Did you know a person is bigger than an egg, chicken egg, bigger than a frog egg, bigger than eukaryotic cells, bigger than prokaryotic cell bacteria, bigger than viruses, bigger than large molecules like proteins, bigger than small molecules like water, bigger than atoms. So you should know the relative rank that I just mentioned. And I guess that's all you need to know. You might want to know what you can see with a microscope, like most cells, eukaryotic, and bacteria. You can see with a light microscope. Viruses are too small to see with the light microscope. You have to have an electron microscope to see them. And that can go down to seeing atoms. And then with our eye, we can't see much smaller than a frog egg. You can see a little bit smaller than a frog egg. And if you look very closely on a watermelon, let's see if I can get that. It's here. If you look closely at a watermelon, you'll see a shape like that in the core of the watermelon, meaning you have a very large piece. There will be little cells like that, smaller than this. Let me see if I can make that smaller. I can't. And you look very closely, you'll see it, especially in the center of the watermelon. I'll try and draw the center of the watermelon. I'm not a very good artist, but I never claimed to be an artist. You'll see these little things right there. Those are actually cells of the watermelon that you can see. You just barely see them with your naked eye. All right, any question about any of that? So what are organic compounds? Organic compounds are typically more complex structurally than an inorganic compound. But organic compounds are held together by carbon. Carbon is important because carbon is an atom that can bind to 4 other atoms. And then organic compounds are held together mostly by covalent chemical bonds holding the atoms together. And if you don't know what a covalent chemical bond is, you'll need to read about that. Inorganic compounds are typically more simple than an organic compound. They typically lack carbon, but they don't always lack carbon. Inorganic compounds are required by living organisms for their growth, their repair, their maintenance, and reproduction. Some examples of an inorganic compound are water, carbon dioxide, and oxygen. Okay, these are all required by living organisms. Water is actually the #1 most common molecule in anything that's alive. It makes up a little bit more than 70% of your body weight, water. And if you notice, carbon dioxide does contain carbon, and it is an inorganic compound. Any question about any of that? Shrink that a little. Let's talk a little bit more about water. Water is extremely important for life, and you'll be doing a worksheet on the lecture on water. Water is a polar molecule, meaning it has a charge, I should say a partial charge. I forget what your textbook calls that. It doesn't call it a partial charge. It calls it a slight charge, meaning it's not a complete -1 charge. It's a partial charge, or your textbook calls it a slight charge. And the partial charge of water is, the negative is on oxygen, and then the hydrogen has a partial positive charge. The reason for this is that water is a polar molecule, and that is the electrons are pulled closer and more will be found more often around oxygen than around hydrogen. And that gives the oxygen a slightly negative charge. And that gives the hydrogen a slightly positive charge. And that makes the molecule polar. This allows water to hydrogen bond with other water molecules or with other polar molecules. And what happens in a hydrogen bond is that the positive charge of hydrogen will be attracted electromagnetically to the partially negative charge of oxygen. And that attraction we call a hydrogen bond. So water has two chemical bonds. It has a covalent bond within the water molecule, and that links the oxygen to the hydrogen. And this is a polar covalent bond because the electrons making the bond spend most of their time around oxygen and less of their time around hydrogen. Anyways, a polar covalent bond is one of the bonds that we find in water. The other bond is hydrogen bonds that we don't find within the water molecule. We find it between the water molecules. Between this water molecule and that water molecule, we find a hydrogen bond. Now the hydrogen bonds are very weak. In liquid water, they break and reform all the time. That's different from the covalent bond, which the bond between the hydrogen and the oxygen almost never breaks. I'm not going to say it never breaks, but most of the time it does not break. Okay, so that's a very strong bond. This is a very weak bond. And it's this very weak bond that breaks and reforms all the time that gives water its liquid properties above 32 degrees and below 32 degrees Celsius, above 32 degrees Celsius and below 100 degrees Celsius. That's what gives water its liquid properties. And that is the molecules break their hydrogen bonds and then reform. And it may reform with this molecule or may form with another water molecule. And that's why water is a liquid. And then it can change its shape because the hydrogen bonds break and reform all the time. Now, if we never broke and reformed the hydrogen bond, meaning that hydrogen bond becomes permanent, it does not break, then we don't have liquid water. We have solid water, and that we call ice. And ice does not change its shape. Any questions about any of that? All right. Water is a good solvent. That means that it can dissolve a lot of substances. All polar molecules and all ionic molecules can dissolve in water. For example, sodium chloride, which is salt, table salt, is an ionic molecule, meaning the chloride has a negative charge and the sodium has a positive charge. It's ionic. This is a -1 and a +1. That's a complete charge, ionic. And what water does is it comes up to the ions in the salt, and then the negative oxygen comes around the positive sodium, and then it will pull that sodium atom out of the crystalline structure, and then that will go into water, and that we have salt water, where the sodium ion is surrounded by negative oxygen, meaning the hydrogens, which are positively charged, are away from the positively charged sodium atom. And then the same with chloride. The negatively charged chloride ion will be surrounded with water, but in this case, the positive hydrogens will orient themselves next to the negative chloride ion and surround the atom and then bring it into solution. And that is how ionic molecules dissolve in water. And then the same way polar molecules dissolve in water. The difference is instead of having a complete positive charge here, we'll have a partial positive charge. And then the negative oxygen will be around the partial positive charge. So polar substances dissociate in water and then form solutes where we'll have the molecules dissolved in the water, the solute. Any question about that? So water is a good solvent. Polar molecules are hydrophilic. That means literally water loving. What it means is just that polar molecules like to be around water and they will dissolve in water because they're hydrophilic, meaning they love being in water. Non-polar molecules like oil are repelled from water, and we use the term hydrophobic. Literally, this means fear of water, fear of fear of water. And that's why when you get oil and water, when you're making your vinegar and you mix it up, they will separate, the oil coming on top, the water and the vinegar below, because the oil is hydrophobic. And literally, it does not dissolve in water and it will separate from water. And all fats, yes, all fats and oils are non-polar and they are, hydrophobic. Things like sugars, ions, they're hydrophilic. Amino acids are hydrophilic. Hydrogen bonding between water molecules gives water some of its properties. And specifically, the hydrogen bond makes water excellent at buffering the temperature. Water absorbs heat in two ways. Most substances, when they absorb heat, they only absorb heat in one way. Like when you heat up a beaker of oil, when you heat the oil, the energy going into the oil will cause the molecules to vibrate more rapidly as the oil starts raising in temperature. And that's actually what the temperature is, the measurement of the vibration of the molecules. The more energy the oil has, the higher the temperature will be, the more the molecules vibrate or move around. Water, on the other hand, can do that, meaning you can raise the temperature of the water, causing the water molecules to move more rapidly and vibrate more, causing the temperature of the water to increase. However, the water absorbs energy in another way, and that is some of the energy is used to break the hydrogen bonds in the water. So each hydrogen bond requires energy to break, and water can take some of the energy you're giving it to break the hydrogen bonds. And so if you give water the same amount of energy as we give oil, and we're talking about the same amount of water and oil, like 100 milliliters, the oil will raise its temperature higher because all of the energy is going into raising the temperature. Whereas water, the temperature of the water and the same energy will not raise as high because some of the energy is going into raising the temperature of the water, and some of the energy is going into breaking the hydrogen bonds. And then as the hydrogen bonds are broken, the molecule breaks off, and then the water molecules that have the most energy, meaning they're the hottest water molecules, they're the water molecules that evaporate because they're no longer connected to the liquid water. Their hydrogen bond is broken, and then they evaporate. And that takes the heat away from the water. So not only is water good at buffering the temperature, water is also good at cooling. And that's why we perspire, to cool our body temperature, which you guys are probably doing that today. It's a warm day today in Western Oregon and Western Washington, and I assume in Eastern Washington and Eastern Oregon as well. I'm not sure about what the temperature is on the coast, but the rest of the state, I think it's pretty warm. Any question about any of that? All right. Water molecules can break apart. I told you that the covalent bond of water hardly ever breaks apart. Well, it can break apart, okay? And water, which is, let me pull up a new file here. Water, which is H2O, and structurally speaking, it's going to be an H connected to an O, connected to an H. These covalent bonds can break. Not very often, unless you're at the temperature of the sun, but they can break apart in liquid water, where when that breaks, let me go ahead and do that chemically. In which case it breaks into H plus OH minus. Let me superscript that. Superscript this. Any question about any of that? This doesn't happen very often. Most of the time, it happens only once in 1 times 10 to the 7th, oops, water molecules. Do you understand that? So if we have 1 times 10 to the 7th water molecules, one of them will break apart like this. Instead of being H2O, it will break into H plus and OH minus. Any questions about any of that? Now the H plus is a proton, and that is a positive 1 there. And that's a negative 1 there. So this is a complete charge. And that we call a proton in the hydroxide ion. This gives charge to water because this water now has a positive charge and that has a negative charge. Whereas H2O, let me back here. If all the molecules were to stay as H2O, the water would have no charge. And if you don't know, distilled water is very lousy at conducting electricity. And that's because most of the water molecules are H2O. They don't conduct electricity very well. They do contract it a little bit because 1 times 10 to the 7th water molecules do break apart into H plus and OH minus. And these, what do you want to call these ions can conduct electricity. When water does break apart, these ions are charged molecules and they can participate in chemical reactions. These charged molecules of water actually give rise to the pH of water. pH literally is the measurement of how many H plus atoms or ions there are in water. And we'll go into that in a little bit. Any questions there? Let's talk just a little bit about the pH. You're seeing a pH scale here. Let me blow that up. So in neutral water, when we first have water, like distilled water, there will be equal numbers of H plus and OH minus, and the pH will be 7. Why is the pH 7? Well, the pH of 7 equals, let me end this here. A pH of 7 means that there's one hydrogen ion in 1 times 10 to the 7th water molecules. I might have a bar there I have to delete. Okay, so that's what a pH of 7 means. pH of 7 means there's one hydrogen ion in 1 times 10 to the 7th water molecules. Any question about any of that? Now, anything below a pH of 7 is an acid. And here we have more hydrogen ions than we have OH ions. So in an acid condition, we have more H plus than OH minus. And a pH of 6 literally means, let me just go ahead and copy this. A pH of 6 literally means that we have one H plus ion and 1 times 10 to the 6 water molecules. Any question there? That's what a pH of 6 means. Meaning the pH is a measure of how many hydrogen ions we have in solution relative to the number of water molecules. And the same thing for a pH of 5 and all the other pHs. When you get down to a pH of 0, let me see if I can do that again. pH of 0 means you have one hydrogen ion and 1 times 10 to the 0 water molecules. 10 to the 0 is 1, meaning pH of 0 means is that all of the water molecules will split into H plus and OH minus. You got that? Which I guess what we can do is right here, this equals 1 H plus and 1. Well, 1 water molecule. So that would be an extreme acid. I guess I don't need to save that. The pH of 0. You almost never see a pH of 0. One of the strongest pHs we have is stomach acid. I think the only thing that's stronger than stomach acid is carbaduric acid, and that's about pH 1. Anything above pH 7 has more OH minus ions than H plus. A pH of 8 follows the same thing. So we're going to go in here. I'll just do it right here. A pH of 8 means we have 1 hydrogen ion in 10 to the 8 water molecules. And that's what a pH of 8 means. Any question about any of that? This is a basic solution where we have more OH minus than we have H plus in solution. And you can get seawater is actually higher than pH 8. Milk of magnesia a little higher than pH 10. Also ammonia. Also bleach is about the strongest pH you probably come across. It's higher than a pH 12. If you ever use oven cleaner, it's actually above pH 13. Okay. And then 14 is the highest basic substance we can get. I'm not sure why it can't go higher than 14, but that's the highest the pH can get. Any question about any of that? All right, that's acid and bases. An acid is a substance that dissociates, giving up H plus in solution. So HCl, if we put it in water, will dissociate into H plus and chloride minus, and that will increase the H plus in solution, making the solution acidic. Why HCl is an acid. A base is a substance that the textbook says dissociates, giving up OH minus in solution. Like when we put sodium hydroxide in water, it'll dissolve into sodium and hydroxide. Hydroxide will increase the hydroxide in solution, making the solution basic. Your textbook does not discuss it. However, a base is a substance that either gives up OH minus, or it is a substance that dissolves H plus in solution. For example, in your blood, we have bicarbonate, HCO3 minus, and it can take H plus and absorb the H plus, reducing the H plus in solution, forming carbonic acid, and that removes the H plus in solution, making the solution basic. So a base is something that both gives off OH minus or it absorbs H plus ion. Okay. And you have to know that from the lesson because your textbook doesn't discuss the second way a base can form. Not sure why the textbook doesn't. I'm not sure. Maybe the textbook author doesn't know, I don't know. The salt is something that disassociates into cations that have a positive charge and anions, something that has a negative charge, but neither of the anion or the cations are H plus or OH minus, because if they're H plus or OH minus, it's not a salt, it's an acid or a base. But if it does disassociate into a cation having a positive charge and an anion having a negative charge, we then call it a salt. The most common salt that you know of is table salt, and that's sodium chloride, which disassociates into Na plus and chloride minus, right there. But that's true for other salts, like magnesium chloride disassociates into magnesium plus, and then 2 chloride ions minus magnesium chloride. There's also a salt. Not table salt, but it's a salt. Okay, salts will give the water the property to conduce electricity, meaning salt water usually conducts electricity, and that we call electrolytes. Sodium chloride are electrolytes. There's a substance that disassociates in water, forming cations and anions, and then can conduct electricity very easily. If you don't know, that's why you should not swim in the ocean during a lightning storm, because if lightning were to hit the water would usually conduct the electricity. Any question about any of that? All right. Organisms need to maintain a fairly constant balance of their cellular acids and their bases to remain healthy. There's actually a term for that you probably learned in Biology 160. You don't need to know it for this class, but that's homeostasis. The pH, as I mentioned, is the amount of H plus in solution. Specifically, it is the measure of the H plus in solution, and you take the log of it and the minus log of it, and that's specifically what the pH is. The pH scale ranges from 0 to 14. It's a log scale, meaning when you change the number by 1, like pH 7 to pH 6, you increase the H plus ion concentration by a factor of 10. You increase the H plus, you increase the acidity, and that lowers the pH from 7, someplace between 0 and 7. On the other hand, if you increase the OH minus or decrease the H plus, that increases the alkalinity, that will raise the pH from 7 to 14. So acidic solutions have the H plus concentration greater than the OH minus concentration, and basic or alkaline solutions, which a basic is the same as an alkaline, have the OH concentration higher than the H plus concentration. Most organisms grow best around pH 7. So like between pH 6 and pH 8. Most organisms like that. The textbook says most organisms grow best between pH 6.5 and pH 8.5, but different textbooks say different ranges. How we maintain our balance, the pH balance, is we have buffers in our body that help maintain our pH. The most common one, I think I actually mentioned it to you. Where do I have that? I guess I mentioned it in the slide someplace. Here, here. It's sodium bicarbonate. I don't remember where I showed you that, but we have sodium bicarbonate in our body, and that helps buffer the pH. So that when you drink a whole container of Coca-Cola, which is acidic, it doesn't kill you because we have buffers in our blood, which, there it is there, like bicarbonate, which helps the blood maintain its pH. Like, for example, if you were to drink a whole bunch of Coca-Cola, this will disassociate. No, this will, because you have acid from the Coca-Cola, it will form with the bicarbonate to make carbonic acid, removing H plus from your blood. So that's a buffer we have in our body and blood to help maintain our pH. Any question about any of that? All right, if not, let's just introduce organic compounds. They are compounds held together by carbon. And here we're looking at some of them. Don't memorize any of them, but all organic compounds are held together by carbon, meaning carbon tends to hold the molecule together. Like right here, all of the links are held together by carbon, except for this right there, which is held together by oxygen. All the other links are held together by carbon. Organic compounds do contain carbon, and they also contain hydrogen. So all organic compounds have carbon and hydrogen. Many of them also contain oxygen. Not all of them, but many of them do contain oxygen. Some of them contain other elements, like many of them contain nitrogen, sulfur, phosphorus. All of these atoms are held together covalently by a covalent chemical bond. This includes amino acids, carbohydrates, nucleotides, lipids, and various hydrocarbons. Compared to inorganic molecules, organic compounds are more complex. They are also capable of more complicated functions. And specifically for biological organic compounds, they have complicated biological functions. What are some examples of organic compounds? Here are some examples of an organic compound. You don't need to know these, but methanol, ethanol, isopropanol. They're all held together by carbon, which can bind to 4 other atoms. Like this carbon is bound to 4 other atoms. The molecule is held together largely by carbon. So carbon is the skeleton or the backbone of the organic compound. And then the organic compound has a functional group, which gives the molecule its property. So the functional group is a group bound to the molecule, which gives this molecule its properties. Like methanol has different properties than methane because methanol has the functional group OH, which gives the methanol different properties than methane. Any question about any of that? Any questions about anything? All right, if not, we'll end the lesson here and I'll see you at 6:30 for the lab.
Thank you.
All right, I'll see you at 6:30.
Is the lab's at 6:30 instead of 7 today?
Oh, wait a minute. Do I have the time wrong? Ah, you're right. I was thinking the class ended at 6:30. It ends at 6:50. Hopefully most of you have not logged off. Because I'm going to continue with the class for 10 more minutes. I got my times wrong. Sorry about that. Are we still sharing the screen?
No.
All right, let me share the screen. I even looked at that and I didn't read it right. All right, let's continue. 10 more minutes. Otherwise, we'll fall behind schedule. The functional groups, they're responsible for most of the chemical and physical properties of the particular organic compounds. Like the hydroxyl group of alcohols gives the molecule its properties and why an alcohol can be attracted to water molecules, whereas methane is not attracted to water molecules and does not dissolve in water. Alcohol, like methanol, is attracted to water and will dissolve in water. It is because of the properties of this functional group. So the functional group helps the methanol dissolve in water. The functional groups that you need to know of are shown in this slide here. Let me blow that up a little bit. And the ones you need to know are in the red box. Okay, so we're looking at different functional groups. And you do need to know what an alcohol group is. It's found in lipids and carbohydrates and in other places as well. You also need to know what an amino group is. It's a group that has NH2 or NH3 on it, and it's found in proteins. You need to know what a carboxyl group is. It is a group that has COOH, and that first O has a double chemical bond. The second one, OH, just has a one chemical bond. This is a carboxyl group. It's found in proteins, lipids, and organic acid. And then phosphate, PO4 with a negative charge, a phosphate group. It's found in ATP and DNA. You need to know these functional groups because these are the functional groups you find in the biological molecules. Okay, so they're commonly found in the biological molecule. You must be familiar with these groups found in the biological molecules. Frequently, an organic chemical compound will have more than one functional group. Like, for example, with amino acids shown below, we have both the amino group attached to the amino acid and their carboxyl group attached to the amino acid. And then this molecule has the properties of an amino group as well as the properties of a carboxyl group. Any question about any of that? So small organic molecules can combine into larger molecules that we call macromolecules. The small organic molecules we call monomers. The large macromolecule we call a polymer. So monomer, one, mer, poly, many, mers. So polymer is made up of many monomers. And a polymer or macromolecule is made up of many small repeating units of the macromolecule. We can make the polymer by combining the monomers by dehydration synthesis, which is also called a condensation reaction, where we pull off an OH group from one monomer and a hydrogen from the other monomer to make water, and then link the monomers directly together. This R being for the monomer and that R from the monomer are then linked together covalently, so the R is bound directly to the R in dehydration synthesis, making the polymer from the monomers. So strictly speaking, if that's a monomer and that's a monomer, this would be a, oh, what is that called? Dropping the word here. Bimer, is that right? Strictly speaking, that's not a polymer, it's only two, but dimer, it's a dimer. Okay. And then we can do the reverse, taking the polymer, combining it with water, splitting the water in hydrolysis to make two monomers. And the water is split, so the hydrogen binds to this R, and then the OH of the water split to bind to this R, and then this OH group binds to that R, making the two monomers. So dehydration is the reverse of hydrolysis, and hydrolysis is the reverse of dehydration synthesis. But dehydration synthesis is the mechanism for how the macromolecules such as carbohydrates, lipids, and proteins are made. And I should mention that the R just stands for some organic group or molecule, and it can be any organic group. Any question about this slide? I should mention to you that you guys are probably familiar with hydrolysis. Remember when Marty McFly told his mother, "Ma, you sure know how to hydrate a pizza." She took this hockey stick of a pizza and put it in her hydrolysis machine, and out came a pizza. And that was hydrolysis, where she made the pizza. Any question about any of that? All right, let's talk about this slide showing you the same thing. Here we have a polymer, and we're going to join a monomer to it. Water will come out in dehydration synthesis, and then we'll link the monomer directly to the polymer, and then water will form. We can then have hydrolysis where we add water to this polymer, and then the water splits apart. The H forms on this part of the polymer, and the OH forms on this part, and we break off the monomer from the polymer. Any question about any of that? All right, if not, I think we'll stop the lesson right here, and we'll continue here next time. Any questions? If there's no questions, I'm going to end the lesson here, and then we will start the lab at 7 o'clock.